Welcome everyone, and thank you for joining us for the keynote speech of the Climate Adaptation Research for BC Agriculture Workshop. This session is the 28th Annual H.R. MacCarthy Lecture in Pest Management. My name is Julie Carrillo, and I'm a Center for Sustainable Food Systems Associate and Assistant Professor from the University of British Columbia. As we begin, I would like to acknowledge that I'm hosting this session today from the traditional, ancestral, and unceded territory of the Hunkaminam-speaking Musqueam people. Each of you are joining us today from many places near and far, and I'd also like to acknowledge the traditional caretakers of those lands. If you don't know whose territories you're on, we'd encourage you to begin that learning journey by doing some research to find out. One way you can do this is by going to nativeland.ca. The purpose of the H.R. MacCarthy Pest Management Lecture is to present an annual lecture by a distinguished pest management scientist or practitioner. The lecture is managed by the H.R. MacCarthy Lecture Committee, consisting of representatives of the University of British Columbia, Simon Fraser University, and Agriculture and Agri-Food Canada. It is funded by revenues from the H.R. MacCarthy Endowment Fund held by Simon Fraser University. This event is presented by the Centre for Sustainable Food Systems at UBC Farm and the BC Food Web. The Centre for Sustainable Food Systems at UBC Farm is a teaching and research center, as well as a local to global food hub working towards a more sustainable food secure future. The mission of the CSFS is to innovate from field to fork to achieve resilient, thriving, and socially just food systems for all. BC Food Web is a web portal project which aims to increase access and connection to current research and other resource materials. In addition, it aims to encourage innovation between farmers and researchers for project collaborations. Today is day one of the annual workshop hosted by the BC Agricultural Climate Adaptation Research Network, or ACARN for short. ACARN is a provincial network launched in 2018 to improve linkages and collaboration to more effectively address climate adaptation research and extension needs in the BC agricultural industry. This workshop is a chance to share knowledge on research and tools available to support agricultural climate change adaptation. It's also an opportunity for questions, discussion, and networking between producers, researchers, policymakers, and industry specialists. It is a place to lay the groundwork for future collaborations in research. Today's webinar topic is Lessons from the Last 10 Years of Drosophila suzukii, Moving Forward with Management. Dr. Hannah Burrack from North Carolina State University will discuss her research on this topic. We will leave about 30 minutes for Q&A after Dr. Burrack's talk. If you have questions for Dr. Burrack, please navigate to the Q&A tab at the bottom of your Zoom control panel and share them with us. Also, feel free to upvote others' questions by clicking on the thumbs up symbol. If you would like the opportunity to chat with other attendees, feel free to introduce yourself and add comments in the chat. Lastly, we would like to acknowledge that these unprecedented times mean we all have new working conditions. Please bear with us and our speakers if there are unexpected distractions or interruptions. Finally, it's my pleasure to introduce Dr. Hannah Burrack, a professor and extension specialist in the Department of Entomology and Plant Pathology at North Carolina State University, where she has served since 2007. Dr. Burrack leads the Specialty Crop IPM Laboratory, which develops biology-based pest and pollinator management strategies. Recently, she has accepted an appointment as the Director of Education and Outreach for the NC State University Plant Sciences Initiative. Dr. Burrack completed her doctoral research at the University of California, Davis in 2007, where she focused on the biology and management of the olive fruit fly, which at the time had recently invaded California. Dr. Burrack carried this interest in invasive species management to NC State and has been a national and international leader in the response to Drosophila suzukii, the spotted winged Drosophila. During her career at NC State, Dr. Burrack has trained 17 graduate students, contributed to an astounding 62 peer-reviewed publications and book chapters, and has generated over $11 million in program funding. In addition, she has engaged with grower stakeholders through contributions to 25 production guides, over 420 meetings to more than 27,000 attendees, and has extensive media contacts. With that, I'd like to hand it over to Dr. Hannah Burrack. You're welcome to begin your presentation.
All right, well, thank you so much, Julie, for the invitation, and thank you so much, everyone, for coming to this afternoon session. I know that presenting and receiving information this way can be a little bit different than what we're used to, but I'm really very appreciative of the opportunity to continue collaborations and conversations. So as Julie said, for the next hour or so, we're going to focus on a discussion of learning from the last 10 years for Drosophila suzukii management. And I say this because we have been living with this organism for now 10 plus years. I also want to point out, though, that the information that I'm going to be sharing is the result of a huge collaborative effort. And so I've been really fortunate to work with a number of wonderful students and collaborators throughout the US and internationally. And I'll share more of their specific names at the end, but I'm highlighting the folks on this first slide who provided some really specific contributions to today's presentation. So a little bit of an overview on the biology and invasion history of Drosophila suzukii, spotted wing Drosophila, or SWD. I'll use all of those names interchangeably throughout this presentation, so they all mean the same thing. And while many of you might be familiar with SWD, some of you may not be as familiar. And so let's spend a little bit of time getting to know the critter we're going to talk about. The males have spots on the end of their wings, which is where they get their common name. The females lack these spots, but instead have a heavily serrated and noticeably larger terminal egg-laying segment in their abdomen, which they use to saw into the surface of soft-skinned fruit and deposit their eggs just underneath. So you can see on this blueberry, respiratory filaments of spotted wing Drosophila eggs sticking out. The eggs then hatch, the larvae feed internally on the fruit, and in many cases, the fruit is otherwise undamaged appearing until some precipitating event results in those larvae being observed. That larval development process takes about a week. Larvae then pupate on, in, or near the surface of the fruit, or they may drop down and pupate into the soil. That entire process takes about 10 to 15 days, and adults can live for 1 month or more under the right conditions. They have no known true diapause, although as others have found, they do change their behavior and their physical appearance in the winter, and so their winter stages can in some cases look physically different than their summer stages. And this video clip is going to illustrate the main concern we have associated with spotted wing Drosophila. This is a video that was taken the first summer that we found spotted wing in North Carolina in 2010 by a research technician in the caneberry breeding program at NC State. And as you'll see, this otherwise sound appearing raspberry had an unexpected surprise present in it. So when Absalom tore this raspberry open, you can see live larvae present here in the center crawling around. And prior to dissecting this fruit, the only other obvious sign that something was going on with it might be a pink receptacle when it was pulled off the plant, or a wet center of that berry. And as you can probably understand, there is zero tolerance for these live larval presence in fruit that is fresh marketed or marketed as whole processed fruit. And this is the crux of the problem for spotted wing Drosophila for us. And spotted wing Drosophila was a truly unexpected invasive species. So this organism was previously known from Eastern Asia, and most notably from Japan, but was not really on anyone's radar screen as a significant potential pest. In 2008, it was detected in California and roughly around the same time in Italy. And it underwent a very rapid, and I would say probably historically unprecedented in recent memory, range expansion for an agricultural pest. In the following 4 years, it was found throughout the United States and also throughout Europe. And this just illustrates that range expansion in the US over time. So from the 2008 detection in California up and down the West Coast to the Eastern US in 2010, and throughout the entire country where fruit are grown. I also wanted to put this range expansion in context with some of the other invasive species that we think about in these and related production systems. So here's our spotted winged Drosophila up here at the top, and that same US map illustrating the rapid expansion and now the locations where spotted winged Drosophila is established and actively damaging fruit in red. You can compare that to Japanese beetle, a foliar feeding polyphagous scarab beetle that was first found in New Jersey in 1916 and has very slowly spread throughout the country. Brown marmorated stink bug had a more rapid range expansion, but has not had the accompanying global range expansion that spotted winged Drosophila has had. The Asian citrus psyllid, while extremely problematic on its host plants, is generally restricted to warm areas where citrus can be grown. Moving on to soybean aphid, found primarily in the upper Midwest and cooler parts of the country since its detection in 2000. And the light brown apple moth, a highly polyphagous caterpillar pest that's a leaf roller, found roughly around the same time in California and in some of the same places as spotted winged Drosophila, but has remained restricted in California. And spotted lanternfly, our new and most recent significant invasive pest of concern, found primarily in the eastern US now, first detected in 2014. And we could add New York up here in pink as well. There have been a couple point detections and some possibly reproducing populations found in that state recently. But I just want to contrast this with the extensive and very rapid spread associated with spotted winged Drosophila. So this really was an intense and very rapid invasion. Illustrating that over time, again found in 2008 in raspberries in California, the initial detection. The following year was found up and down the west coast of North America, and then into the southern US and the Midwest, and then throughout New England, the Mid-Atlantic in 2011. And by 2012, it was detected everywhere in the United States where we grow fruit. Prior to its detection in California, it was really only a significant concern in a small area of Japan around the turn of the 1900s. It was noted as a pest of cherries. The solution to its pest status was to stop growing cherries in those areas. It was found in Hawaii in surveillance programs targeting native Drosophila species to determine non-target effects of fruit fly management programs to fruited fruit fly management programs, but it doesn't cause any significant damage in Hawaii. And then that simultaneous spread and detection in Europe in the same time period, finally moving into South America in 2014 in both Brazil and Argentina and Chile in 2015. So really the only berry producing region of the world that's been spared. Colonization by suzukii has been Australia. And responding to an unexpected invader comes with a host of challenges. So there's a typical process that we tend to move through when we detect an invasive species. That remains the same, but the information gaps and resource needs tend to be much greater when we're dealing with an organism that wasn't on our radar screen to be preventatively monitoring for. So when you detect a new invasive species, you first try to delineate its range. Where is it found? And how far has it spread? In order to do that, you need effective identification methods, monitoring tools, and you also want to understand the population genetics of the organism you're working with and potential invasion pathways. You next want to identify hosts and use that information to quantify risks. So you need to know what hosts are affected, what hosts are preferred and perhaps at higher risk, the timing of infestation, host selection, as well as economic thresholds that might be associated. And I'll tell you right now, going back to that video I showed you, our economic threshold is essentially 0 for these guys. So we don't have an effective economic threshold in most cases. Next, we focus on developing short-term management tactics. These are tactics that can mitigate those losses and reduce the risk that we identify. And that typically centers around pesticide efficacy, developing rotational programs for pesticides, monitoring and mitigating any potential pesticide resistance, and assessing whether there are natural enemies present that might be benefiting our potential control of these organisms. Next, those short-term management strategies almost always come with unintended consequences. And so we have to mitigate the effects of those short-term management strategies, which have a reliance on pesticides, and develop long-term strategies. This includes determining non-target effects of pesticides, non-chemical control tactics, and ultimately, in many cases, developing classical biological control. Finally, and what we often spend our last phase of responding to invasive species focusing on is trying to understand the broader impacts outside of agricultural systems. And this is the direction the talk will move into toward the end of the slides, because that's also what we're starting to think about in my group. So where are they outside of agricultural systems? Are there effective monitoring tools that can be utilized in those habitats? And how are non-crop hosts playing a role in contributing to significance? These are the areas in bold and underlined that my group has focused on the most. And so this is the areas that I'll be focusing on as we move through the remainder of these slides. So first, let's talk a little bit about invasion routes. So when we think about delineating the range of these organisms, how are they getting here? There's been several population genetic studies focused on spotted wing Drosophila. So this is one of the first ones that I was involved in. A group out of NC State led by Nadia Singh and her student Jeffrey Adrian, focused on understanding the relationship between populations that were collected from around the world, Japan, a number of locations in the US, as well as Spain and Europe, and found that there really wasn't a clear pattern in relationship between these populations. There were Spanish populations that seemed relatively distinct, but then among all of our other populations, there was a whole lot of overlap. There was a similar study that was done focusing on the incipient populations in Ukraine immediately after the detection of spotted wing Drosophila there. And what they found was evidence of recurrent and multiple introductions from multiple different locations in Europe and abroad in this early development of an infestation. And this is really consistent with our understanding of the population genetics of spotted wing as we move forward. We likely think that introductions are coming from multiple places into new areas, and that there are recurrent spread and flux within those established populations. Joanna Chu at UC Davis, as part of a large collaborative USDA grant that I've been leading, has also conducted extensive genetic analyses. She collected populations of spotted wing Drosophila from 40 areas in Asia, Europe, and the United States in order to try to understand to a greater extent the population genetics of this organism. And she did whole genome sequencing and looked at a range of different PCA analyses to try to determine the relationship of populations. And so the Asian populations segregate fairly well. Populations in the Eastern US and the Western US separate from each other. And so there's a distinct Eastern and Western US population. And then populations in Japan separate from those in Europe. So what it appears is though that our populations in the US are different from the populations that are present in Europe, which are different from the populations that are present in Asia. And we have two separate population groups in the Eastern United States and the Western United States. But within both of those regions, we have migration over time throughout the year. And so this means that when we have undesirable traits for things like insecticide resistance, there's the potential that can rapidly spread through these populations over these broad geographic areas. And that's how we're going to be utilizing some of this information moving forward. When we think about delineating range of a new invasive species, one of the first questions we ask is how do we effectively monitor them? There's been significant research focused on monitoring spotted wing Drosophila, but this has been a really tricky challenge. The challenge is not finding an attractive lure. Sugar and yeast fermentation lures are very attractive. Volatiles associated with fermentation or just straight sugar and yeast as a coarse product are highly attractive to spotted wing Drosophila. They are more attractive than any other lure combination, even pure compounds. However, they're not specific to spotted wing Drosophila. So you get lots of bycatch associated with these fermentation-based lures. So you can catch a lot of flies, but they become a tricky trap to utilize. And what I'm showing you here are two figures, both in season and off season, from Cesar Rodriguez Seona's group. He's based at Rutgers University, and he has really led a lot of the monitoring work that we've been focusing on in our large collaborative project. And this figure just illustrates that lack of specificity. So this is a figure from work that I led comparing attractants across a range of different states, 9 different states in the US, and looking at apple cider vinegar, that yeast and sugar slurry, another permutation of a active fermenting cup, and some commercially formulated these trace lures. But across the board, the proportion of flies that were spotted wing Drosophila per week was very low. So less than 40% in most cases of the flies were suzukii. When we approached about 50% suzukii as compared to other Drosophila species, was in this synthetic lure that we compared, and it was overall a less attractive bait than the other ones. So in addition to being not terribly selective, the monitoring tools that we have available to us also attract flies that are in some cases qualitatively different than flies that are found attacking fruit. So they may not be monitoring the organisms that the individuals of the organism that we are most interested in tracking. And so this is from that same environmental entomology study where we had that multi-state comparison. This was work that Doug McPhee, an undergrad at the time in my lab, took on. And he dissected spermathecae from at least 10 flies per trap per week for each of these different trap types. He dissected ovaries from flies from each of these different trap types per week, and determined the number of mature eggs, those with fully formed respiratory filaments, that were present. And what he saw is that prior to fruit being susceptible to infestation, so weeks 1 and 2, the number of mature eggs per fly were much higher than when fruit was susceptible to infestation. So the number of mature eggs per fly in the traps dropped down once there was attractive fruit able to be infested, although the number of flies that were being captured across all of those trap types continued to increase during that period. Similarly, this is work done by my graduate student, Katie Swoboda-Badarai, where she looked at egg load in flies that were captured either on the surface of traps, inside of traps, meaning they entered the trap, or on fruit near those traps. And what she found over the course of 3 years was that when we collected flies from fruit, they consistently had more eggs in their ovaries than the flies that were entering the traps. So in other words, what we see in the traps is a representation of reproductively immature females, whereas the flies that we're collecting from fruit are more likely to be reproductively mature females capable of attacking those fruit. And so we're really not getting a complete picture from these traps in terms of the biology of the flies, although we may catch a lot of them. And if we catch a lot of them, we're also catching a lot of their friends and their cousins in there as well. So it's a tricky nut to crack in terms of having a more specific lure that we can make decisions about in terms of either presence or absence or management decisions. However, in some instances, monitoring has proven useful for management. So this is modeling done by economist Miguel Gomez, who's based at Cornell University. He collaborated with our partners at University of Maine, Frank Drummond and others, to develop this Excel-based interactive management tool for lowbush blueberries specifically. And it incorporates the value of the crop, the cost of the pesticide that a grower might be considering using, and then the male trap capture that they're obtaining in monitoring traps. And it takes all of this information into account and makes a recommendation to growers as to what would make more sense to harvest now, to make an insecticide application, and then they can decide which of those tactics is most advisable. This was work done based on lots of monitoring that Frank had conducted to demonstrate that trap capture can correlate to infestation risk in this system. But one of the challenges in generalizing this beyond Maine is they have a very focused and short production period that's later in the growing season. And so they are less likely to have continued prolonged infestation, such as some of our other crops might experience. But we don't want to throw traps out with the bathwater entirely, because they do, in some of these narrow, more focused situations, potentially inform our management recommendations. All right, so moving on to host risk. So we've talked about our monitoring tools that we have available and our known population genetics. What about hosts? Who's the most at risk? And across the board, Rosaceae appear to be preferred crop hosts. Among those, raspberries are the most highly preferred. When we presented a set volume of fruit in the laboratory to female flies, they attacked raspberries most aggressively, even when they had a choice between other fruit. Strawberries were the second most infested, blackberries the third, and blueberries the least most infested. This is consistent with work by a number of other folks looking at the hierarchy of preference in this fly. And in addition to being more preferred, more flies will survive in, will develop faster, and can tolerate greater degrees of competition than in raspberry, as opposed to other fruit diets. And so it's not only a more attractive host to them, their progeny also do better in raspberries. Those fruit are going to become susceptible to infestation when they begin to ripen. And so this is work that Katie did again, focusing on trying to determine under field conditions when are fruit most likely going to be damaged. And she caged fruit immediately after bloom, after pollination had occurred, to exclude fly egg laying, then held them in cages until they reached the desired ripeness stage, and then exposed them to an ambient population of flies. And so at our research station location, where we had very high populations of flies, the fruit were unmanaged, unharvested, and high trap captures of spotted wing. Infestation occurred as soon as that fruit started to change color. So as soon as that fruit had a little bit of a blush on it, flies were potentially laying eggs in it. They laid significantly more eggs in fruit as they continued to ripen from red to purple and to perfectly ripe, but the potential for infestation was there at the time of initial color change. Katie repeated this work at a commercial field the following year. This was a location that was actively managed, where the grower was picking the fruit on a frequent basis, making insecticide applications, and therefore had relatively low numbers of Drosophila that he was capturing in traps. And in this case, she saw no infestation in the green-pink fruit, very low levels of infestation in red and purple fruit, and infestation really focused and concentrated on those ripe fruit. So this is the model of what we think is happening. They prefer, under an ideal situation, to infest ripe fruit. But when population densities are high, infestation will then spill over into less desirable hosts, purple fruit, red fruit, and then to green-pink fruit. And we can see this play out in this time series data as well. So as fly populations increase in the top figure, we see infestation begin first to show up in our ripe fruit, next to develop in our purple fruit, and then to move into our red and green-pink fruit right around the same time. And so you see that temporal relationship mirror what we would expect to see happening based on this spillover effect. There are other factors that also influence host risk. So we know plant phenology impacts host risk. This is a schematic I used to illustrate that for North Carolina, where I'm located. The yellow line illustrates trap captures at a single location in Western North Carolina, and the colored bars indicate when our different host crops are present and harvested over that same time period. So in North Carolina, strawberries are harvested early in the spring, and we're usually done picking them by June. In many cases, while we have flies present this entire time, we catch flies all winter long in North Carolina under the right conditions, their numbers are low enough that strawberries don't experience detectable infestation. Blueberries are at this inflection point where our populations generally begin to increase rapidly at the start of July. Some blueberry varieties are harvested earlier than that, while others may fall into this more risky period. And so variety within a blueberry can determine how significantly it will be impacted by suzukii. And our blackberries and raspberries in North Carolina are harvested generally from midsummer all the way into the fall if we're thinking about primocane fruiting varieties. And those are exposed to the highest risk of spotted wing infestation based on population density. And they are also the most preferred hosts, so they are at most risk with respect to preference. We also know that firmness influences host selection. So this likely plays into selection among ripe and unripe fruit. But even within individual fruit, flies will prefer in a no choice scenario, less firm fruit, as well as in a choice scenario. And finally, what else is present in the area will determine infestation. Not only will it determine infestation between an individual fly making a choice, but as illustrated here with work done by my postdoc Lauren Diepenbrock, who is now at University of Florida, it also influences what the next generation might do. And she took flies that she had reared from blackberry and exposed them to blackberry again, or pokeweed, which is a weedy non-crop host of spotted wing in North Carolina. And they had a 50-50 preference for those two hosts. They didn't indicate any preference for one or the other. Whereas flies that had developed in pokeweed had a strong preference to lay their eggs in blackberry following developing in that suboptimal non-crop host. And so what that the previous generation develops in may also predispose it to make a different host selection and perhaps predispose it to move into a crop host from a non-crop host. So co-occurring habitat also influences preference. Those non-crop hosts that spotted wing can utilize are extremely diverse. And so this is just a figure pulled out of a study I really like out of Europe, where they looked at a range of different crop species, non-crop hosts, and overlaid the period in which they are ripe. And this just very elegantly illustrates that we essentially have a non-crop host present for spotted wing all year round. And the same phenology co-occurs in North America as well. So maybe not the same species, but we have this same potential availability when the environmental conditions are right to have something that these flies could potentially eat. In addition to non-crop fruit hosts, folks have also observed spotted wing Drosophila feeding on things like mushrooms, flowers, mammal and bird manure, and decomposing fruits and vegetables. So there's nothing special about fruit that they require in order to survive and thrive. They can also utilize a range of different non-crop hosts. They may face more competition from other Drosophila species in those non-crop environments, but it doesn't mean that they can't eke through and maintain a population in that habitat. So just because there is no fruit available does not mean that flies have nothing to eat. Moving on to talk about short-term management now. As I indicated, typically when we think about short-term management of invasive species, we are focusing on insecticide-based control. And Phil Fanning, who is now based at University of Maine, updated this figure that we had originally developed several years ago, looking at efficacy of different pesticides against spotted wing Drosophila. This is a survey-based rating, so he surveyed entomologists across 9 different states with 19 and different state and crop combinations. So the states that were included here illustrated above. And what you're seeing is that for the most part, our most effective modes of action include pyrethroid insecticides, spinosad insecticides, 1-carbamate and lanate, and then some organophosphate insecticides. The diamides, which include vertiprin, Exoril, are also reasonably effective. So we have 4 classes, 5 if you throw lanate in, of insecticides that have some degree of acceptable control against spotted wing Drosophila. We have a relatively full toolbox of insecticides. However, we have some constraints to using that relatively full toolbox. One is pre-harvest interval. So a material has to have a pre-harvest interval that is short enough to allow its use during the susceptible fruit infestation period, meaning it has to be something you can use while the fruit is ripening and ripe. It also secondarily has to be effective. We can't use it if it doesn't have a short enough pre-harvest interval. We won't use it if it's not effective. For a large number of growers, export tolerances and residue levels are also a significant concern. Folks in my lab, as well as other programs, have demonstrated that, as we would expect, if you use these materials in accordance with their label, you do not exceed residue constraints in the country in which you are growing your fruit in. However, our residue levels differ between our trading partners, and so that can make marketing that fruit internationally more challenging. And then finally, resistance management. We want to have effective rotational programs between these effective active ingredients. We don't want to focus on a single mode of action because the rapid development time of spotted wing can result in resistance development. And this is something we are actively monitoring and concerned about. So this again is work as part of this collaborative project with a number of different cooperators. And what I'm showing you here are data generated by Brian Gress and Frank Zalem at UC Davis, where they looked at Spinosad resistance that has developed in California populations of spotted wing Drosophila. The susceptible population in all of these figures is the solid line, and the resistant population is the finer dashed line. And you can see in all cases, you have a significant level of resistance in this population that has developed to Spinosad. They are also seeing similar concerning signs associated with zeta cypermethrin, which is the active ingredient in Mustang and Mustang Max. All of this work has been done associated with a rapid bioassay that we've used to develop screening for resistant populations as part of this project, headed by Steve Van Timmeren, Rufus Isaacs at Michigan State University, and Ash Sayal, who's based down at University of Georgia. So moving on to our long-term management, we know that there's these associated risks with these short-term management tactics, our chemical control tools. So what non-chemical management tactics have been developed? So here on the next couple slides, I'm going to be showing you work that has been coordinated by an organic focused project led by Ash Sayal out of the University of Georgia with collaborators at the institutions shown across the top, including us at NC State. So we've demonstrated that pruning can reduce infestation in caneberries. Importantly, it does not have the same reduction in blueberries. So what I'm showing you here is just spotted wing Drosophila per gram of fruit. When you have low pruning or high pruning and low canopy density, that can significantly reduce infestation as compared to high canopy density with low pruning rates. But in blueberries, we did not see that similar effect. What's responsible for this reduction in infestation appears to be a change in the canopy climate. So it makes it slightly warmer and also reduces the amount of relative humidity present. And so over those two interactions, the reduction was 0.14 fewer larvae per gram of fruit in that low density canopy. We also know that frequent harvest reduces infestation in caneberries. This is work that was conducted by Heather Leach at Michigan State University, where she harvested raspberries either every day, every 2 days, or every 3 days. And those every day and every 2 day harvest intervals, she significantly reduced the number of eggs, small larvae, and large larvae. There was a little bit more flux in the second year, but again, there was a significant reduction in small larvae and large larvae over that more frequent harvest interval. And this ties hand in hand with our third non-chemical management tactic, which is focused on cold storage. So we know from work done in my laboratory that post-harvest cold storage stops development of spotted wing Drosophila if you put it into cold temperatures. In this case, I'm showing you 35 degrees Fahrenheit as opposed to room temperature for 3 days. It extends the development time by 3 days. And so you essentially stop whatever life stage development that fruit goes into cold storage at. And this is true at temperatures from 41 degrees Fahrenheit and cooler. When we collect fruit from the field with a mixed infestation of larvae in it, we also see a significant reduction in survival when fruit is held at either 0 degrees Celsius or 2.2 degrees Celsius for 3 or more days. That effect, however, is substrate dependent, and that's important to note. So you can see that we saw a significant reduction from fruit that was held at room temperature after collection, regardless of host, but we saw lower survival in blueberries, strawberries, and raspberries than we did in our blackberries. And so we saw a greater degree of survival in our blackberry, and we never were able to reach 0 survival in that substrate. So cold storage is helpful, but it's not sufficient in and of itself. What is going to be most beneficial is a thorough frequent harvest schedule that allows you to reduce the number of immature larvae or eggs that might potentially be in fruit, and then apply post-harvest cold storage treatment that further reduces the number that's present. Finally, in terms of long-term management, there's been work developing genetic control tactics. There's 3 different genetically based strategies that are under development. First is the development of a genetically sterile spotted wing Drosophila strain. These have been developed at NC State as well as at other institutions. This would be a version of the sterile insect technique in which you would release sterile male spotted wing Drosophila in large numbers to reduce the populations. The difference between traditional sterile insect technique and genetically sterile flies is that genetically sterile flies would not require chemosterilization or radiation-based sterilization before released. So you would reduce those possible risks in producing those flies. There are also gene-drive spotted wing Drosophila under development and testing. And these would be flies that are engineered to carry a gene that makes them weaker and then pass along that gene to flies that they mate with. And there are a couple different modes of this method of control under development and testing. And then finally, RNAi targeting spotted wing Drosophila. This is where something else, perhaps a yeast as a food source, would be engineered to carry RNAi, which would make spotted wing Drosophila weaker or kill them. It would be a biological pesticide. And then flies would be exposed to this species-specific RNAi, and that would ultimately reduce the population. What my group has been focused on over the last year or so has been trying to assess the social implications of these genetic pest management tactics. We think that that's particularly important if we consider these novel and innovative strategies for pest control to also be thinking holistically about how best to implement them once they are ready for field utilization. And so what we've been doing is surveying interested spotted wing Drosophila stakeholders via an online survey. We had a total of 185 respondents split between academia, growers, non-governmental organizations, industry members, and regulators or government officials. And this is of a total of about 681 surveys that we sent out. This interested group of stakeholders is broadly supportive of the idea of gene drive, for example, against spotted wing Drosophila. Blue areas indicate a degree of support for this tactic among these interested stakeholders. However, despite this high degree of support, there was significant concern about possible adverse effects of gene drive. And so these are just some preliminary data. This is work that we are in the process, hopefully in the next week or two, of submitting for publication. But what I'm showing you here are respondents who were in orange, concerned, extremely concerned, highly concerned, or moderately concerned. They expressed some degree of concern about a given possible negative outcome, or in blue, were not concerned about those outcomes. And in the center column is indicating whether those respondents were supportive of gene drives overall, neither supportive or opposed, And you can see even among the respondents who supported genetic control tactics, such as gene drive, there was still a high degree of concern about some possible adverse outcomes that could be associated with these different tactics. So we think this is important to tease out. It helps us understand what best to evaluate prior to the release of a novel genetic control tactic. It also helps us inform how we conduct risk assessments in the future around these and other tools. So just to summarize a bit of what we've learned about spotted wing Drosophila in the last 10 plus years. We've learned that risk varies greatly between crops and regions. We've learned that our non-chemical tactics are important, but they're never going to be sufficient for control in high-risk crops. We will likely always have a need in some of our most heavily impacted crops for some additional management. Our naturally occurring biological controls are insignificant. While I didn't show you the data on that, I will show you a link to a webinar to get more information there. And these management goals will require long-term efforts. I want to spend the last few minutes here talking about some of these broader questions associated with ecosystem health that we've been starting to explore. And this is work conducted by my graduate student, Joanna Elsenson, who just defended her dissertation about a week and a half ago. And so this is fairly new work that is just making its way into review. And she was interested in whether or not these agricultural pests like spotted wing invade remote and natural areas. This isn't something we normally look for agricultural pests. And there's really been pretty limited work in this area. And it's been focused more on pests that feed on trees, things like spotted lanternfly, ambrosia beetles, and thrips. And so she wanted to determine whether spotted wing behavior changes in a natural setting versus a commercial setting. So she selected locations in Western North Carolina that were isolated. She determined whether they were isolated or not based on these light map data. And so what I'm showing you here are locations where she sampled in 2017, 2017 and 2018, and contained either wild grown caneberries. She sampled exclusively in blackberries in the open circles, or cultivated caneberries. And so she had research station locations in 2017 down near Asheville, North Carolina, and up here near Boone, North Carolina in 2018. Those were our cultivated sites. And then all the other locations were the wild locations. In 2017, she was focused on breadth, and she wanted to understand how widespread spotted wing Drosophila were in some of these natural areas. So she had 4 locations in and near national forest land, non-commercial land, and those 2 research stations. She sampled greater than 1 kilometer apart in order to get as much breadth as possible within these locations. She sampled fruit at 5 ripeness stages, 20 berries per stage when available at green, blush, red, purple, and ripe. And only ripe were sampled in the cultivated samples in 2017. That's not the case in 2018. And collected the number of eggs that were present per berry in each of the fruit that she sampled. In 2018, she had 1 forested site where she sampled along an elevational gradient in order to understand what role elevation might be playing in infestation, and 1 research station. After 2017, she was very confident that spotted wing was everywhere she was looking for it. So the breadth of samples were no longer quite as important. She was more interested in drilling deep into the local dynamics of a smaller number of sites. Again, up to 20 berries per stage for these different ripenesses, and the number of spotted wing eggs per berry. When combining all of the data from all those locations in years, she found spotted wing Drosophila eggs at all sites and all sampling locations. She found higher numbers of berries in cultivated fruit. However, when you determined the number of eggs per gram, there were higher numbers of eggs per gram in wild fruit. And those wild underripe stages were more heavily infested than the cultivated underripe stages. And I'll show you a little bit more of why we think that is on the next slide. It's important to think about the dynamics of size in these observations because a cultivated berry is anywhere from twice to 5 times as large as a wild blackberry. And so when we look at just a per berry basis, we're not necessarily getting an accurate representation of how flies are utilizing this resource. So this is the same figure, same information illustrated over time. So you have cultivated on the left and wild grown fruit on the right. And what you'll see first is that our cultivated graphs are much more orderly. So these are eggs per gram across the top, and then we have percent infestation for any given ripeness stage across the bottom. And you have this very clear progression of infestation, as I previously showed you, with respect to the least ripe fruit being least infested, next ripeness stage more infested, and then our perfectly ripe fruit is the highest rate of infestation. And you can see the percentage infestation mirrors that. So lower percentage infestation in the blush fruit and similar percentage infestation as you increase ripeness stages. For our wild grown fruit, it's a much messier story. So what you have there is a lot of overlap between the different ripeness stages in terms of the number of eggs per gram, as well as the percent of fruit that's infested. And when we think about interpreting this, one thing we need to understand is that we have selected our cultivated fruit to present itself in this clear timed ripeness. And so we expect, in order for fruit to be harvested in an orderly fashion, for it to ripen in that progression. Whereas wild grown fruit, you are more likely to have green fruit next to red fruit, next to pink fruit on adjacent bushes and plants. And so the resource availability is much more variable in these wild fruit than in the cultivated fruit. One thing Joanna also noticed when she was collecting these samples is the progression from unripe to ripe is much quicker in the wild grown fruit. It's a much faster time period. And so the reward for a fly infesting an underripe berry is perhaps greater in the wild grown fruit than it would be in the cultivated fruit. So next, Joanna wanted to understand the difference between, if there was any difference between preference in wild and cultivated hosts. And so she's not the only person who's looked at this, but I think her results are really interesting in comparison with some of the other studies that have looked at wild and cultivated hosts. So she went out and excluded infestation after petal fall, after the flowers had been pollinated. And then she collected them when they were ripe on the plant and returned them to the laboratory and conducted a laboratory bioassay with our colony where paired females were allowed to lay eggs for 90 minutes in either an equal weight of berries or an equal number of berries. And so this is what that looked like. So you either had one tiny wild berry, one large cultivated berry, or several tiny wild berries equivalent in mass to the cultivated berry. When there were equal weights, so when they had the same volume of fruit available to them, the flies strongly preferred the wild grown fruit. When there were equal numbers of fruit, they had one wild fruit and one cultivated fruit, they strongly preferred the cultivated fruit. So again, this apparency and resource availability is important to consider, but when the resources were equal in volume, there was a strong preference for the wild grown fruit. The last project I wanted to briefly highlight is work that's being conducted by my PhD student, Laura Kraft. This is one of Laura's projects in addition to the cold storage work that I mentioned earlier. And she's been focused on developing means to detect what flies have fed on following collection. And so we've been focused on determining resource use in flies to see if they are utilizing fruit as an adult resource. And so she's successfully developed primers for blueberry and blackberry. We had a goal of developing primers for raspberry and strawberry, but it is very difficult to differentiate between rosaceae. And so we have a primer we're comfortable with blueberries, a primer we're comfortable with blackberries, but we can't go down any deeper into the rosaceae there. So that was a very tricky task for her to undertake. It's something that will probably be easier in about 3 or 4 years because there's a huge rubus sequencing initiative going on here in the Research Triangle Park area in North Carolina. So we were probably just 5 years ahead of where we would be to make this a much easier project. And then she determined how effective these primers would be for detecting a single feeding event over time in a series of laboratory experiments as well as field experiments, with the goal of discovering what adults had fed on. And she also tried to determine whether larval food resources would be detected within adult guts. And so Laura's in the process of writing all of this up, and this is a teaser for that paper when it comes out into the world. I'm going to go ahead and wrap up on these last couple of slides, and I just want to highlight again the large number of folks who've contributed to this work, as well as the other projects going on in my lab, as well as the collaborators both at North Carolina State University, other institutions, and the support associated with these projects. Much of this work that I shared with you is a product of a USDA NIFA Specialty Crops Research Initiative funded grant, which had collaborators at all of these institutions illustrated across the bottom. Another output of that project has been the production of annual webinars that have been focused on different topics associated with spotted wing Drosophila. So you can see here the link to those webinars. These are our 3 topics to date. And fortuitously, we have another webinar tomorrow where we are drilling down even deeper into the topic that I'm talking about today. Today, I really focused on work that was associated with my lab group. Tomorrow, we're going to be taking a broader look at products associated with the entire project for the last 10 years of spotted wing Drosophila management. I will go ahead and pop this link in the chat box when we are done and in the Q&A session. But I wanted to encourage you guys to check that out as well. And with that, I am going to stop talking and open this up to any questions that you guys have.
Great, thank you so much, Dr. Burrack, for that wonderful talk. We have several questions in the Q&A. So what I'll do is I'll go through the questions and moderate. And if people have additional questions, please feel free to post them in the Q&A now, and we will hopefully get to all of those.
And I'm going to go ahead and stop sharing if that's all right.
Sounds good. I should also note that we are recording the webinar, and we will record the Q&A session as well and provide that afterwards to participants. So our first question is from Dr. Paul Abram at Agassiz, Agriculture and Agri-Food Canada. Paul says, excellent talk so far, Dr. Burrack. I was wondering, what have you been able to find out about SWD's pest status in its area of origin in Asia? For me, reliable information or data on this has been hard to find. So could you point us to some specific resources?
Sure. So Jana Lee, who is with USDA ARS based near Oregon State University, did some excellent work at the really early stages of spotted wing Drosophila invasion in the US. And she translated a number of Japanese articles from, or she and collaborators translated a number of Japanese articles from that 1900s period. So from 1916 through about 1935-ish or so, describing the impacts in cherries and blueberries in Japan. And that's where I had extracted that phrase from, which is they basically stopped growing cherries in some of the more heavily impacted areas. In other parts of Asia, so Korea and Eastern China, up until the global range expansion for spotted wing, it did not seem to be a pest of concern. And it's a little bit tricky to figure out why that might be, but I think part of it might be there wasn't a lot of like commercial cultivation of the crops that they really like in those parts of the country. So the blackberry industry in Korea is pretty nascent and young. Similarly in Eastern China, it's also a pretty young industry. There's also a fairly young blueberry production industry there. There's a lot of wild rubus in that part of the world. And so I think that's probably where spotted wing was happiest. But I think cultivated rubus was pretty uncommon. That's my guess, is that the things that they really love to eat were not being grown extensively until the last 20 years or so. And so perhaps there wasn't that same cultivated host relationship as they were fortunate to stumble into in their introduced range. And most of us in that 1900s era Japan series of papers also think that that's probably an invasion as well, right? So the natal populations, or the truly native populations, are probably more Eastern China up along the Himalayas, moving into cooler parts of Central Asia. And those are probably that true native range. And that Japanese event, where there was all of a sudden this really significant number of papers being produced about damage in cherries and blueberries and other things, is probably also the result of an introduction there. What seems to have happened over time in Japan, at least, is that they are elevationally restricted, kind of like what we see in Hawaii. So in Hawaii, they're found at higher elevations where crops that they might feed on aren't grown. And that also seems to be the case, at least in some parts of Japan now. So they're found in wild cherries, but there's a strong elevational effect on their densities in those wild cherry systems.
Thanks, and we'll see if Paul has a follow-up to that. Right now, I'm going to move on to a question from Kira Jack. And she asks, what is known about the mechanisms controlling differences in susceptibility across different fruits? So why does blueberry seem to be less susceptible? Is this due to skin thickness, or is this due to something else?
It's a really good question. I think that blueberry is less susceptible partially because it is harder for them to lay eggs in. So we do see that really clear relationship between fruit firmness and preference. And we also can modify substrates and give them the same quality of substrate that's softer or firmer, and we will see that same relationship. But I also think it's not as good a host for them. So if we infest blueberries at the same rate as we infest raspberries, for example, in the lab, we will get more surviving pupae out of a raspberry than we will out of a blueberry. So I think there's probably a nutritional component as well as the physical components that make it less attractive. I'm not saying blueberries are resistant to spotted wing. They're not. If they're present in the field and there's enough flies there, they will just as happily feed and reproduce in blueberries as they will in other hosts. But when they have a choice, it's not going to be necessarily their favorite thing. And we see this here in North Carolina because a really common weed in some of our blueberry fields are wild blackberries or wild brambles. And we will always first detect infestation in those wild caneberries or wild brambles than we will inside the blueberry field, even if they are co-occurring and ripe at the same time. So that preference plays out in field situations similar to what we would predict based on what we see in the laboratory. So I think physical characteristics play a role. I also think that there's probably some nutritional component as well, just given that we see lower survival and performance of those progeny once they're laid inside the blueberry.
Great, thank you. Let me go to the next question. This question is from Jen McFarlane from ES Crop Consult. Regarding eggs being laid in red or green-pink fruit when SWD populations are high, are the larvae actually surviving in those fruit and completing their life cycle?
They can survive. It's similar to what I just mentioned with blueberries, that we see lower survival rates coming out of underripe fruit than we do in ripe fruit. And so it's a lower quality source, we think, than the perfectly ripe, ready to pick fruit would be. But they can survive. We will get larvae pupating out of a blush pink blackberry if there's a high enough infestation level for them to be there.
Mm-hmm. So your results show that even after cold storage, spotted wing Drosophila is developing in these fruits for market. So we have a question from Maria L. about whether there are other quality control options specifically for raspberry that reduce these levels. And how concerned should consumers be about spotted wing Drosophila in fruit for consumption in terms of their own health and safety?
Yes, and so I'll tackle that last one. Yep, I'll tackle that last one first, which is, I would not, first of all, because we have zero tolerance for spotted wing Drosophila infestation at the producer level, meaning if a grower picks fruit and it goes to a wholesaler, and they inspect that clamshell and find one larva, they will likely reject the entire load of fruit from that grower. And so the risk for damage typically resides at the grower level. They're the ones at the most risk of experiencing a negative outcome due to infestation, meaning rejection of a given harvest. So the folks who sell you your berries have been trying very hard to make sure that nothing detectable will make it through to a consumer. So I think we can be very confident in that we are not buying a whole lot, if any, spotted wing Drosophila at the grocery store. Similarly, more direct market folks are also cognizant of spotted wing and trying to make sure that what they market to their consumers is as low risk of infestation as possible. That being said, that possibility exists. And even if you were, say, walking out in my research plots, I'll throw myself under the bus. If you were walking out in my research plots where I don't spray for spotted wing because I want them there, I still eat my berries. One of the great joys of being an entomologist working in small fruit crops is that you get to eat your untreated controls, right? And I'm not going to give that up just because there might be a fly in them. And they will not hurt you. They are not poisonous. They cause no, human impact other than being unsightly. So that risk is low. And further, the folks who are selling you your berries are doing their darndest to make sure that nothing is getting through. And so they're really absorbing a lot of that risk of potential loss and trying to make sure that no infestation sneaks its way on through. As far as other control options, once you have picked the fruit and if you are concerned about possible infestation. So cold storage is important. Pre-harvest, that frequent harvesting is really important. So making sure that what you get off the plant, if you're harvesting every other day, you're probably only getting eggs if anything is there, because it takes 1 to 2 days for those eggs to hatch. And so if you have the tiniest guy possible, the least amount of damage is potentially occurring, you get it in cold storage. It's going to be most susceptible to cold storage when it goes in there quickly. And then if you are still concerned about possible risk, there are other post-harvest management tools that have been developed for quarantine type scenarios. Those include things like acceptable fumigation tools when they are labeled for a crop. And other scientists focused on quarantine management, so shipping of fruit to other countries who may not want to receive spotted wing, have developed some irradiation protocols as well. So those are other post-harvest strategies that are effective, but for a local consumption would probably be unnecessary.
Yeah, so I have a follow-up question to that, and I see we have lots of other questions too. So given this, essentially zero economic threshold, and the prevalence of spraying to control spotted wing Drosophila, how do you approach collaborative research with growers when you really want to do monitoring in fields yet spraying is ongoing? So how can you effectively evaluate other management techniques in these actively sprayed fields?
Yeah, it is really challenging. And so we have had, we've had 2 different strategies for 2 different sets of growers. We have had, for our blueberry growers who are growing a lower risk crop at a time of year when our populations are lower, they are willing to let us do have untreated controls in their farm. So we've been able to have untreated controls in our large scale on-farm blueberry work, where we can do monitoring or where we can compare to treatments directly in that same location. For our caneberry growers who are growing the most susceptible crop at the riskiest time of year, we typically compare to an industry standard. So we'll have an industry standard control practice or a grower standard control practice that will compare whatever treatments we are assessing next to that. If we are interested in natural population dynamics, then we will always have a paired research station location where we can have a truly untreated scenario, but we can't go to our blackberry growers and say, give us an untreated control, because we all know what will happen if we do that. It's not as risky for, say, a blueberry grower, or in some cases a strawberry grower, where they may not experience any damage and then we have to jump through the hoops to try to get data because we don't have pressure. So then we end up doing like simultaneous lab experiments with things to have some sort of data come out of it, but at least the grower is experiencing less risk. So that's what we end up doing for our caneberry growers. For better or for worse, we've never been to a blackberry farm that has like effectively eliminated spotted wing Drosophila presence. And so even if we are artificially manipulating the population dynamics to some extent, we still find flies there. We still find infestation there. And so we are able to detect differences in most cases. And as I showed in some of the work that Katie did, you can actually sometimes see more subtle differences when you've artificially lowered the population with some pesticide usage that might be just overwhelmed when you have that naturally occurring population. And I didn't show these data, but Joanna Elsenson, my PhD student who just finished, had a whole series of experiments looking at female host choice and how that was influenced by fly density and by infestation status of the substrates that they were choosing between. And what she found is that you can't run those experiments for more than like 2 hours, and the differences will go away. And so some of these choices that the insects are making are really fast and kind of subtle. And so it's going to be almost impossible to observe that in a high pressure situation in the field where you just have so many flies present. And so it took a lot of her optimization to figure out those experiments to really measure those behaviors. But that's the unintended benefit of having growers use pesticide, is sometimes you can see these subtle changes that big populations overwhelm.
Our next question is about oviposition deterring pheromones from Andrew Edmonds. So is there an ODP pheromone for this pest already known?
So there is not a quantified oviposition deterring pheromone yet. So there's evidence that there might be something. This is work that folks in Von Walton's lab have done. There's folks in Europe who have also worked on this. And then Joanna, who I just mentioned, has done some of this work as well, looking at the behavior associated with infested and uninfested substrates. And it does look like they are doing something and that it has some impact on female host choice, but what the chemical actually is and how that plays out in the field is still unclear. It's not the same as other Drosophila species. So other Drosophila species have more of a local aggregation pheromone that they produce because they benefit from being grouped together. They can more effectively utilize a resource when you have lots of, say, Drosophila melanogaster, our genetics lab rat. They like being together. Spotted wing does not. And so they don't utilize that, the chemical that other Drosophila species utilize to mark their food resources.
So kind of along that same vein, we have a question from Warren Wong, who's an MSc student at SFU about to defend next week. And Warren asks that given that it has been found that SWD is attracted to yeast and vinegar are reproductively immature, is there current research being conducted on other host cues for gravid females? And do you have thoughts on their foraging behavior or the cues that they're using?
Yeah, so Cesar Rodriguez-Sona has done a really nice series of work trying to tease out fruit volatiles and leaf-associated volatiles to see if plant-associated chemicals could be more effective in luring in the flies that are qualitatively damaging fruit, so flies that have mature eggs and are mated. They are just far less attractive even to those flies than these fermentation compounds. So food seems to be a much bigger motivator. That's what we think they're cueing in on in these fermentation odors, is that they're cueing in on an adult food source rather than a larval substrate. And so that seems to be just a bigger motivator in the environment than those, from an odor standpoint, the larval substrate that they might be laying their eggs in. What I think is happening, this is my opinion, I think that visual cues are probably playing a really big role in where the females go. I think they are probably keying in on large apparent fruit and then interacting with it physically when they land on it. So testing it out with their ovipositor and then palpating it to see what it tastes like, maybe other sensory structures on other parts of the body. But I think that the visual cue draws them in, and then the physical interaction with the substrate determines when they're going to lay an egg or how many eggs they're going to lay in that area. That's my guess. And so I don't know that volatile cues are going to be our biggest help there. There has been work by Jana Lee at USDA, as well as other folks looking at the role of color in attraction for flies. So can you have different colored traps and pull flies in that way? And again, those fermentation-based food lures are just such a much louder signal than even the color. So you can have these unbaited color traps or color traps with the fermentation trap, and you can get a little more in a red trap baited with a fermentation lure. But it's probably not going to make that much of a difference in your monitoring your population dynamics.
So we have a lot of people who have the same question as Rosina, which is whether integrated livestock such as chickens could make an impact on spotted-winged Drosophila levels.
So that is a really good question. And I don't know if anyone has, that is one area I'm not sure if anyone has looked at. I know that for codling moth and Pomocurculia, there's been work on things like hogs and other livestock moving through apple orchards. But I'm not sure if the same work has been done on spotted wing. It would be interesting to see how if that could decrease be part of an integrated cultural control practice in decreasing populations in small scale areas. I will say adults are very mobile. And so you could potentially decrease the population that is coming from your planting by having some local orchard or vineyard or berry field floor management. But you're still probably going to have populations that are coming in from the non-crop hosts and surrounding areas. And one set of data I didn't have time to talk about was this very common observation that we always catch more flies on the edges of fields than we do in the interior of fields. Even in an unmanaged scenario, we catch more flies along the edge. And we think that's because they are probably not spending all of their time in our crop fields. They're probably spending time in our sheltered, non-crop adjacent habitat and then moving into our crop fields to lay eggs, mate, and interact with each other that way. And so you may still have populations in the ambient area that are impacting your planting, even if you got rid of every fly that originated from the berries in your field.
Mm-hmm. We have a question from Bonnie Zand about whether you have any thoughts on what caused the explosion of spotted-winged Drosophila from 2008 onward.
It's a really good question, and I wanted to do a little more digging before this talk, and I just didn't have time, but I feel like that means something had to change, right? And so the things we think about when we think about these big population explosions of something that has been hanging out somewhere quietly, like in Hawaii, for a couple of decades without causing any meaningful problems. We think about things like environmental change. So did something happen in the climate that precipitated a explosion of a just simmering along population? We think about trade. So normally we get our bugs where we get our stuff from. And did something change with respect to trade routes around that time period that resulted in introductions? Because again, remember, this was not just North America, this was Europe at the same time with which look to be different flies because they appear to be genetically different from each other. So my money is probably on trade. Something probably happened trade-wise that resulted in more flies or different flies being moved more places. So that's my suspicion. And I do wonder about increases in crop production in their native range and what those patterns look like, or purchasing of crops from other parts of the country, other parts of the world to their native range, like increase in consumption of things like caneberries or vaccinium or strawberries in parts of the world where they were native to, and if that precipitated some increased movement. So that's my guess, is that something changed with respect to trade, and I want to do a little more digging and see what that might be, but that's my suspicion. Just given the really rapid change on a global basis, it seems less likely to be like a climatic event, because that's not going to be so uniform in all of those places.
So can you address, kind of given the seasonal aspects of spotted wing Drosophila activity, what's really driving that in terms of reproduction and just different patterns? Is it host plants? Is it microhabitat? Is it climate changes in alternative hosts, or what?
Yeah, so I think pretty much everywhere where they occur, they go through a winter bottleneck. They like moderate temperatures. They're surprisingly not as happy in South Georgia and Florida as they are in North Carolina and a bit further north. So they probably go through a pretty strong winter bottleneck in those areas. Even though we can catch them all winter here in North Carolina, our trap captures go way down in the winter, and they become inactive when it's cold. And places north of us, that bottleneck is even stronger. I don't think they're going locally extinct in, say, New York or Ontario, but I think that their populations get really small and take a while to build back up. So I think that's the biggest population synchronizer, is that bottleneck they go through in winter. And then in spring, they need some fruit host in order to really ramp up their population. While they can survive on non-fruit hosts, they're in competition with other Drosophila species. So that's probably not going to be as effective a resource for them to build up their populations in. And so they're going to be eking along in whatever non-crop hosts they can find or overwintered populations until they've got the first fruit that come out in the springtime. For us, crop-wise here, that's strawberries. I think strawberries are probably a big driver of initial population buildup. We don't have crop loss in our strawberries. I very rarely get calls about spotted wing Drosophila in spring fruiting strawberries, but I think in culled fruit, in fruit at the end of harvest, after they've stopped picking those fields, I think that's a really good toehold for them to really build up their populations and turn a generation or two. And then I think crop phenology is going to drive a lot more of subsequent populations. So what is in the area that they can continue to reproduce on?
We have a question from Tracy Hoopel-Schuster from the BC Ministry of Ag.
Hi, Tracy.
You talked about the cold storage options and some fumigation, but Tracy asks, are there post-harvest treatments such as modified atmosphere being tried out currently, such as used for apple storage?
Yeah, so Peter Follett, who is another USDA ARS scientist, has done some work on both controlled atmosphere and irradiation. He's done CA in combination with irradiation. There hasn't been, at least not published work that I can find, on just MA modified atmosphere or CA controlled atmosphere by themselves. Most has been focused on irradiation and fumigation. I'm sure that you could develop effective ways of managing that, but then it's just dealing with the substrate, because you've got these very different substrates, and so you probably need to have work done in each of them to make sure you're not having deleterious substrate effects, because that's the tricky thing with all these post-harvest management strategies, is substrates differ in how they tolerate them. So I'm not aware of published work on just modified atmosphere or just controlled atmosphere, but Peter has done some stuff in combination with that and irradiation.
So we have a question again about cold storage, and knowing that all of these different options can lead to resistance developing, do you see any evidence for resistance to cold storage or cold tolerance increasing with these practices?
That is a really good question. I'm not sure they are done that cold storage is a consistent enough practice across all of the crops that we would at this point have selected for tolerance to that, or increased diapause trigger or something. It's not beyond the pale. Bugs are smart. They figure out ways of developing resistance to things we don't think that they can develop resistance to. But I don't think the selection pressure is strong enough right now, at least for cold storage, because we do have, it's not at the producer level super widely adopted. Where it's happening is mostly at the wholesaler level. So at your fruit purchaser level, the name you see on the box that you buy at the grocery store, those folks are the ones that have the best cold chain. And those are also the entities that are not accepting fruit that has detectable infestation at the time of delivery. And so they're selecting out any fruit that has infestation that they're concerned about before it makes it into their cold storage chain. So there's not as much cold storage happening at the farmer level as there is at the wholesaler level. So I think the pressure for resistance at this point is probably pretty low.
We have time for just one more question. And we have a question from Allison Fox about this preference for wild fruit, and particularly for the wild fruit that's growing along the field margins. Would you give the suggestion to growers to ever plant wild fruit along these margins as a way to pull spotted wing out of the crop? Or is this, do you think this will serve just to increase population levels?
Probably the latter. We have a lot of conversations about things like trap crops, because when we talk to people and we say they love raspberries, and people are like, great, can I just plant raspberries next to my blueberries and I don't have to spray them? And unfortunately, all of the berries are tricky to manage. And so getting something that's going to fruit when you want it to fruit and then be able to destroy it so it's not causing some additional problem, to me seems like it's probably more of a management challenge and would require a lot of fine tuning and optimization than it might potentially be worth. So I think that's the tricky part with thinking of a trap crop scenario. The other question we get relative to what's happening on the field margin is, okay, do I just go in and take out all those non-crop hosts? And then my question is always, well, how far can you go? Because these flies are so mobile, folks will say, I can go 50 feet into the woods and I'll take out all the blackberries, or I'll go 100 feet into the woods. And it's like, okay, well, what about 101 feet? I don't have enough science at this point to be able to say how far away you want to be from that non-crop host habitat to result in a meaningful reduction in infestation. And so that is something that it's not easy to recommend at this point.
Especially since we know those alternative habitats can have a lot of important ecosystem services beyond pest control.
Exactly. So you're going to mess up your pollination by taking out all of that non-crop habitat, and that might be equivalent or worse than getting rid of spotted wing.
So thank you so much, Dr. Burrack. I know we have not had a chance to answer everyone's questions today, so I encourage all the attendees to log on to today's workshop discussion board on Padlet. Link is in the chat and on the Eventbrite event page. And there you can continue the conversation with Dr. Burrack and with others that have joined the webinar. We'll follow up after the workshop with a session recording and a roll-up of the resources mentioned today. And I just want to thank Dr. Hannah Burrack. So if we could all give her a virtual round of applause, that would be fantastic. And also behind the scenes, we'd like to thank the ACARN Workshop Planning Committee, UBC IT, and the workshop funders, and the BC Ministry of Agriculture and other donors. So finally, let us know what you think about the webinar. You'll be directed to a 2-question survey about this session after the webinar, and we'd really appreciate your feedback. So with that, we are done for today. Thanks, Hannah. Oh, sorry, we are not done for today. There were lots of questions about natural enemies, so I encourage you to go to the next session where there'll be talk about spotted wing Drosophila, new parasitoids in BC by Paul Abram and Chandra Moffat, and also by Von Walton. So hope to see you there. All right, thanks all.